The overall goal of the serial block phase scanning electron microscopy technique is to analyze mitochondrial morphology, structure, distribution, volume, and number in a defined region of the mouse brain.
This method can help answer many key questions in neurobiology, such as whether changes in brain mitochondria, structure, number, and distribution contribute substantially to neurodegenerative and neurodevelopmental disorders. The main advantage of this technique is that it automates the process of imaging serial sections by incorporating a custom ultramicrotome built into the low vacuum scanning electron microscope chamber.
To begin the experiment, wash the previously prepared glutaraldehyde-fixed tissues 3 times for 5 minutes each in 0.1 molar cacodylate buffer. Post-fix the tissues with 1 milliliter of cacodylate buffered 0.1% tannic acid for 30 minutes at room temperature. After post-fixing, wash the tissues in cacodylate buffer. Dissolve 0.3 grams of potassium ferrocyanide and 0.86 grams of sodium cacodylate in 10 milliliters of distilled water. Just before use, add 10 milliliters of 4% osmium tetroxide and stain the tissues with 2% osmium ferrocyanide solution for 90 minutes on ice. After staining, wash the tissue in distilled water. Treat the samples with freshly prepared 1% thiocarbohydrazide or TCH solution for 20 minutes at room temperature. Then wash the samples. Next, stain the tissues with 2%